Okay, so introducing our next set of panelists will be personalgenomes.org board member Misha Engrist.
Thank you. So I'm going to introduce Randy Buckner, who I met about 10 minutes ago. He is principal investigator of the Cognitive Neuroscience Laboratory and professor of psychology at Harvard University and a member of the Center for Brain Science, where he is affiliated with the Athenula A. Martinos Center for Biomedical Imaging at the Massachusetts General Hospital, Harvard Medical School, and an investigator for the Howard Hughes Medical Institute. His research applies neuroimaging techniques to explore brain areas involved in human memory. In true PGP style, he is a self-experimenter. In 2008, he told science writer Carl Zimmer that he had already scanned his own brain dozens of times, something he started doing at age 20 when he was designing experiments for his PhD at Washington University in St. Louis. By the age of 38, he said, "I can already see the difference between my brain now and when I started more than 15 years ago." And so now he is going to tell us about genes and brains in a talk entitled Genome Meet Brain Connectome. Randy Buckner.
So it's a pleasure to be here among the self-explored. You may be looking at the most studied head on the planet, or one of 5 or 6. I've been in this scanner hundreds of times. I've had a number of different kinds of scans, molecular scans with PET, every kind of fMRI. I've gone into an MRI scanner for 5 hours straight on a bite bar to decide that's not something we should ever subject another person to. And all of this is in the service of trying to understand what I think was one of the more difficult challenges, and something that we're only beginning to make headway on, is connecting our genome with the kind of behaviors we exhibit, both in terms of the normal individual typical variation that all of us have, but also when it leads to mental illness or risk for mental illness. And between those two things, our genes and the behaviors and our traits, is our brain, made up of over 100 billion neurons with trillions of connections. And everything we do, everything we think, all our behaviors are mediated through this matrix of connections, and now information through neural activity cascades through this incredible wiring. And so what we're trying to do is develop tools to actually measure this brain connectome, the wiring diagram of the brain, and also the functional interactions that ultimately are the foundation for behaviors. And I want to tell you a little bit about that today. So the kind of measurements we make are shown up here. This is a scan. Several of you in this room may have actually contributed to this image here. This is a scan that we take in about 12 minutes, which is the white matter, the major white matter bundles that connect the brain. This is not the brain connectome. And when I say the connectome, I mean all of the connections, the individual synapses that weave together this trillion connections across 100 billion neurons. What this is the major highways, and reflects kind of the state of the art today. And just to demystify some of these technologies, and is there a timer should be starting soon? Just to demystify these technologies for you, this actually takes advantage of just a kind of a lucky trick that MR physicists can exploit, and that is that water diffuses differently depending on whether it's in white matter pathways or not. You know this. If I take a speck of dye and I dropped it in a bucket of water, you'd see the dye diffuse rapidly in all directions evenly. If I took the same speck of dye and dropped it on a celery stalk, you'd see it move along the stalk but not against it. The white matter bundles in the brain are much like celery stalks, and if we take an MRI scanner and we tweak it to measure water diffusion in hundreds of different directions, we'd actually make images such as this, which are very gross and very crude estimates of the large-scale projections across the brain. We and others across the field are pushing the limits of these technologies as quickly as we can. One example of this is just shown here. Just to give you an example, this is sometimes affectionately known as the brain bucket. But this is a homemade device, an insert that goes into an MRI scanner that's 64 individual coils. Some of these are up to 128 individual coils. And all they do is just through engineering increase the sensitivity and help us to get better and better pictures of brain structure and function and connectivity. The state of the art can be seen here in this kind of image. Now keep in mind, these are commonplace images. We know we can go into a hospital and get an image of your brain structure, but if you step back from it, it still is quite remarkable. Without a scalpel, without touching an individual, you can beam out remarkable detailed information about brain structure. And here you can see with modern scanners, you could zoom in and see exquisite detail in the matter of minutes with this kind of engineering technology. Now, we are not at the level of the Connectome per se. This is a microscopic image made by Jeff Lichtman and colleagues, Josh Sains, using a technique referred to as the rainbow mouse, where they got individual neurons and transgenic mouse line to fluoresce with different colors so they can actually track in exquisite detail the connections amongst different neurons. And here is an image, an extraordinary image of the connectivity in just a very small microscopic area of the brain. We are not on the horizon to be able to make these kind of measurements in humans, but we're able to see gross architecture and organization of large-scale circuits. And here's just an example of that. I'm referring to this as a functional connectome. This uses another kind of technique that is now widely available for measuring architecture of the human brain, and now is becoming more sensitive so that you can actually measure the architecture in quite a bit of detail in an individual person's brain. The technique takes advantage of another fortuitous quirk of nature, and that is that brain regions that are connected to one another tend to fluctuate their activity together, even when you're not doing anything, spontaneously. In fact, most of the activity in your brain all of the time appears to be unrelated to the things that you're seeing or you're experiencing in the world around you, or the actual things you're doing. It tends to be intrinsic, spontaneous. It may relate to internal mentation, but we actually think most of it is at a much lower level than that. There's just an enormous amount of intrinsic activity, and critically, this intrinsic activity is correlating across areas that are connected. So what that means is you can have somebody lie in an MRI scanner and over 10 or 12 minutes just measure the spontaneous activity of their brain and get an estimate of the connectional architecture of their brain. And I'm showing you one example of that here, where I've just flattened out the cortex. Now, as you know, the brain is highly gyrified, and it's very actually difficult to make sense of that kind of complex anatomy. So what we do is we inflate it a bit, much like taking the raisin and inflating it back into the grape, so that you can see all the areas on the surface. And that's what you're seeing here. And you're seeing actually the functional connectivity of a small region placed here in association cortex in the parietal lobe. This is the front of the head, the back of the head. These are the regions in the visual cortex that you see with. Many people heuristically refer to the prefrontal cortex over here as the regions, the critical regions for thought and contemplation. Now what you're seeing here is the connectivity, the functional connectivity of this region in parietal cortex. And what I'm going to do is I'm going to actually animate it so that moves across the cortex so you can see the different connectivity profiles as you go across even small distances across the cortical mantle. And so this is the complex functional architecture of the average brain, where you can see that each of these nodes across the cortex are interconnected to a large distributed set of regions. And as you move across even small different distances on the cortex, you can see that there's markedly different connectivity patterns. And in ways we still don't fully understand, we think that this complex architecture, these patterns of connectivity is what allows us to do different kinds of functions, and we're learning about the specialization of these systems and also how these different networks work together in the service of coordinated thoughts and behavior, and also how they're becoming disrupted in illness. And just to give you one little vignette, a story, I just want to mention a little bit about what we know about one behavioral trait. It is actually one of the more dominant traits that you can see if you measure behavior across individuals, and it has to do with emotionality. And generally, in the technical term, we often call this trait negative affect or negative affectivity. And it broadly relates to how anxious an individual is versus how laid back. It tends to cluster, so if you make measurements, you'll tend to see at a behavioral level that this trait will correlate. You'll tend to find, and if we go around this room, you'll find a lot of variation. Some people will tend to have a bit more anxiety, a little bit more negative affect would be described in lay terms as being a little bit more on the neurotic side. This, in many ways, is a good thing. These traits correlate with productivity, and in fact, many of us believe that they're actually selected for evolutionarily because they're beneficial to fitness and behavior. But when they become too extreme, they can lead to anxiety and depression, and so many of us have tried to understand these systems. And we know a bit about some of the networks that contribute to this trait variance. For example, we know that there's a critical structure buried deep within the brain, here shown here in the amygdala, that's involved in different affective dimensions. If you show individuals who are lying in a scanner various kinds of emotive stimuli such as despair, or one great trigger for this system is emotional faces. Emotional faces are quite striking. You could just look at an emotional face and it just grabs your attention. There are many ideas about why it does so. One, it's another human being, which is something that's an important cue to us. But the other thing about a fearful face is it tells you there's something dangerous in the environment, but it doesn't tell you what it is. Alfred Hitchcock knew this in his movies. He clipped to the individual's face, not to the thing attacking first. And that's because he intuitively knew that a feel for face is an uncertain cue that drives these visceral systems. Well, 50 or 60 years later, we started to anchor off of this. And for example, if you have somebody lying in a scanner, you present these kind of stimuli, you'll see heightened activity in the amygdala during this kind of presentation of fearful faces. And in individuals who are the most anxious, have the highest negative affect, you'll actually tend to see heightened responses in the system, reflecting that there is some trait variation that you can capture in the brain. And we think that this kind of variation is both a product of our genes and the environment, like everything else. And in the brain, the brain by definition, especially when we get out to these higher order systems, is a highly plastic system that develops throughout infancy and childhood, but continues to modify itself through learning and experience. So anything in the brain, to some degree, is going to be an intimate interplay between the genes that pull the levers and tendencies of how these circuits are going to wire, but then also the experiences that impinge upon them to get them to wire the way they do in an individual. And there was a really remarkable series of studies done by Jerome Kagan more than 30 years ago, where he did an interesting thing. He took very, very small toddlers, sometimes children in their infancy, but typically around 1 or 2 years of age, and he simply measured their responses in novel environments. One simple test that was along these lines, but there's a more elaborate series of novel experiences that were given in the laboratory. But one thing, for example, is if you take a 1-year-old and you shake your keys in front of the 1-year-old, you'll see a child will respond to this kind of novel stimulus. And if you look at a large number of children, you'll find that some become very excitable or inhibited by this kind of event. The novelty is an extreme stimulus. And what he did is he came up with another number of metrics to actually measure the variability of temperament in toddlers and infants. And then asks, does that predict anything as you go on? And what's remarkable is that if you come back to these now teenagers or young adults who had been in these studies, what you can find is that their traits at these very early ages were to some degree predictive of whether they'd be highly social or have social anxiety, for example, as young adults. And this suggests that either in the environment, but more likely biology at a very early age is already pushing and pulling individuals in different directions that their experience in the world will play out. And Carl Schwartz actually did a very interesting study where he brought back the individuals who had been tested in infants, now as young adults, into the scanner, and he found that the responses of these toddlers predicted heightened activity in this emotional system as young adults. And that's quite remarkable, showing us how important these links between genes and biology is, with very important traits such as how socially interactive we're going to be as an adult, although mediated through a complex series of steps and environmental influences throughout our lives. And we know a little bit about the details of this anatomy now. We know that this emotional system, the amygdala, is interconnected. And here's one result from a macaque brain showing the projections of this system in the prefrontal cortex, particularly along the medial prefrontal cortex along the midline. And you can see here in black, these are the regions where you see projections between these limbic emotional structures buried deep within the brain and these prefrontal regions. And while this is an oversimplification, one general model that we think of is that these systems are kind of in a push-pull relationship with each other. These emotional systems are driving affect, are driving behaviors through the kind of emotional and salience that they provide. But these prefrontal systems, these control systems, are kind of tempering them and controlling them and mitigating their overreactive responses. And one model for this is given here. We think a little bit of the emotional systems as being like the stirrups that drive a horse forward, such as Napoleon here going into battle, and the reins as being these prefrontal control systems that pull the system back and guide it. And in a healthy system, you want both of these things. You want our emotions to drive and motivate you forward, but also these control systems to have the right balance and interplay to actually control them and use them most effectively. And so one question we had is, do these traits that we see vary across individuals, how do they relate to these systems, and can we connect them to genetics? And we did something that is not terribly unlike, I think, the kind of group study that many of you are involved in, is that we took advantage of ongoing research and kind of built on it and built on it and built on it. We actually called this project the Superstruct Project. Not many people know what the word superstruct means. You can look it up. It is actually a word. Most people know the related word construct. But superstruct specifically means to build over or upon another structure, to erect upon a foundation. And what we realized is there was thousands upon thousands of research scans going through Boston and tens of thousands across the United States every year. And so what we did is we approached individual investigators and we built on these imaging protocols. We engineered them to be as fast as we could to get structure and function in a useful way across individuals. And then we just tagged them on across 20 or so investigators now, and we're able to get thousands of individuals in a rapid time. We're able to get about 3,000 individuals going through this kind of study to get brain structure, behavior, and now genetics. And a number of you I know contributed last year during the conference, actually came and participated in this study. And so we could start to look, what are the biological substrates that mediate this kind of system? And just to give you one very brief tidbit, you can actually see it. If you measure the trait, negative affect, emotional affect, heightened anxiety, and this is normal, typical variation, not clinical variation, what you find is that as an individual has increased trait anxiety, you see actually at a structural level an enlarged amygdala, very slight, and you actually see thinning of the projection zones of the amygdala in the prefrontal cortex, suggesting that there might even be a push-pull by which some individuals have effectively, we're guessing, a larger contribution of prefrontal control, and other individuals have a larger contribution of these affective systems. And this is not to say one variation of the trait is good or bad. Individuals who differ along these dimensions will have different personalities and different skills. In some contexts, one extreme will be better equipped for a task, and in another context, other extremes. But interestingly, if you look at the upper fifth of individuals, you take the top 20% of so are individuals on this behavioral trait, you can actually see a negative relationship between the size of the amygdala and these medial prefrontal control regions. And this does not capture information on the individual, but it suggests that these kind of behavioral traits can be seen in the structure and functional organization of the brain. Now, we're just starting just now to have large enough samples to explore what are the genetic contributions to this circuitry that I just showed you. And the first hints are coming out. And one initial finding that we just had a simple question. So here we're looking at typical variation, the kind of more laid back versus more anxious variance that all of us experience even within our own families or across our friends. Very typical variation. So what we took is the genetic combinations of genetic variations that led to risk for anxiety and depression and asked, are those same variations what contributes to normal variations across all of us in these systems and circuitry? And what we found is that while it captures a very small portion of the variance, there is a relationship. So the genes and variations that contribute to risk for depression, which are the bars shown over here, also relate to these brain structures that I showed you as covariating with this trait, this negative affect trait. Interestingly, it was specific, that these particular structures didn't covary with genetic variations that were risk for schizophrenia or bipolar disorder. And so in addition to showing that there is some meat connecting the biology to the variation of these brain circuits, it also suggests a hint that some of these relationships might be specific. And this is what we're now heading for, is to try to understand at a more mechanistic level specific genetic variations or pathways that might be contributing in ways that we could make these links. And so I just wanted to end by showing you how we're moving towards this kind of connection to biology. Thank you.
Okay, we have just 2 minutes for questions. If you can raise your hand if anybody has a question for Randy. Here we go. Glenn, when you're done, pass it back to me, please.
Is there any evidence from functional MRI that ApoE4 homozygotes think different?
A great question. As many of you or all of you know, there's a genetic risk for Alzheimer's disease, a protein polymorphism carried by 2 SNPs that has 3 variants of the protein, E2, E3, and E4. Double E4 places you at the highest risk for Alzheimer's disease, double E2 at the lowest risk. And so here's an interesting question. Is there a variation that would reflect a developmental advantage? Is that the question that you would see, that there are hints of an advantage at younger ages for the double E4? To my knowledge, I don't know of one. We've actually gone after this. One of our motivations, because I work in the Alzheimer disease arena, in collecting this large sample, was to actually ask, is the ApoE risk for Alzheimer's disease mediated through how it affects the structure and organization of the brain at much younger ages? And we have not found evidence for that.
Randy, it was a remarkable presentation. Just one question for next year: are there going to be more opportunities for getting personal genome project participants into the fMRI machines?
So if people are interested in that, absolutely. So if a show of hands, who would be interested in popping over for an hour into a scanner? There will be. Thank you.